Dear students, this is my 12th class on renal system. Today I am going to discuss about renal regulation of acid-base balance. How kidney regulate acid-base balance. As a result of metabolism of non-volatile acids and volatile acids are formed in our body. These non-volatile acids include inorganic acids such as sulfuric acid and organic acids such as lactic acid, keto acids, and also include volatile acids. Volatile acid is carbonic acids. So activity of all enzymes in our body are regulated by H plus concentration. Precise regulation of H plus concentration is essential for proper functioning of our body. pH of extracellular fluid is maintained at a very narrow range, that is 7.35 to 7.45. This is the normal pH from 7.35 to 7.45. If it is more than 7.45, it is called alkalemia, and if it is above 7.8, it may lead to death. And if it is below 7.35, it is called acidemia, and if it decreases below 6.8, it may lead to death. So, to achieve H plus homeostasis, there should be a balance between H plus production or intake and its removal from our body. So there should be a balance between H plus production and its removal. How body keep blood pH in a normal range? There are three systems. I think you have already studied about this. One is buffers, body buffers, then role of lung and role of kidney. We are going to discuss now. Okay. Chemical buffering. Buffers, they are very fast in action. You might have studied about bicarbonate buffer, phosphate buffer, ammonia buffer. So they are called chemical buffering, and they decrease maintain pH between 7.35 to 7.45. Then regulation of PCO2 by lung. Lung mainly regulate by regulating partial pressure of carbon dioxide. If more amount of carbon dioxide, it stimulate respiratory center and there will be hyperventilation and carbon dioxide washout and pH is maintained. Okay, and if PCO2 is less, respiratory center is less stimulated. Okay, now regulation of bicarbonate by kidney. Kidney regulate bicarbonate reabsorption. Okay, so these are the three mechanisms: role of kidney, role of lung, and role of buffers. And buffers we have already studied: bicarbonate buffer system, phosphate buffer system, and ammonia buffer system. These are three main buffer systems in our blood. Now, role of kidney. So we are going to discuss the role of kidney. Kidney regulate H plus concentration in ECF mainly by secretion of H plus. Secretion of H plus, otherwise called acidification of urine. Urine become acidic. So excess H plus is secreted or excreted in urine, and urine become acidic, and this decreases H plus load in our body in the ECF. Then reabsorption of bicarbonate. It regulates reabsorption of bicarbonate. In case of alkalosis, bicarbonate reabsorption will be less. In case of acidosis, more bicarbonate is reabsorbed. Both secretion of H plus and reabsorption of bicarbonate takes together. So secretion of H plus or acidification of urine occurs in all part of our renal tubule. So we already discussed different mechanism of H plus secretion in our body in the renal tubule, and it occurs all part of our renal tubule except thin ascending limb and descending limb of loop of Henle. Secretion of H plus, sodium H plus counter transport or sodium H plus antiport. It is an example of secondary active transport. Sodium H plus counter transport, it occurs in PCT, thick ascending limb of loop of Henle, early distal tubule. So this is sodium H plus antiport or sodium H plus counter transport. It is secondary active transport. In the basolateral membrane, there is sodium potassium ATPase. It produces electrical gradient for sodium, and down the electrical gradient, sodium is passively reabsorbed and H plus is secreted. That is sodium H plus antiport or sodium H plus counter transport. And sodium H plus counter transport in early part of PCT is associated with the bicarbonate reabsorption. Okay, so there will be filtered bicarbonate in the renal tubule. So this H plus combined with the filtered bicarbonate, as a result carbonic acid is formed, and this carbonic acid again split into CO2 and H2O, and it is diffused into the renal tubular cell, and inside the tubular cell it is again unite to form carbonic acid. In presence of carbonic anhydrase, it again split into H plus and HCO3 minus. H plus is secreted and HCO3 is taken into the renal tubular interstitium. Okay, so bicarbonate reabsorb along with H plus secretion. Now, another mechanism is primary active transport. It is occurs in intercalated cells of renal tubule, collecting duct. Okay, late DCT and collecting duct. So there is H plus transporting ATPase. So H plus sodium H plus ATPase or sodium potassium H plus ATPase or sodium H plus ATPase in the collecting duct, the tubular lumen. Okay, so this is an primary active transport. Energy is utilized in these steps, and a new bicarbonate is formed. Okay. So pH of glomerular filtrate is 7.4. It will be equal to pH of blood, and pH of urine, it is 6. It is the average pH of urine. This means that there is net tubular acidification. So glomerular filtrate is 7.4 and pH of urine it is 6. So this means that there is net acidification. And this is the minimum pH possible, that is 4.5. Okay. So what is pH? pH is negative logarithm of hydrogen ion concentration. So pH of the extracellular fluid is 7.4 and pH of minimum possible pH of urine is 4.5. So what is the difference in H plus concentration, H plus ion concentration? So in the renal tubule there may be 1000 times more H plus ion concentration will be there. Okay, and this minimum pH is maintained by the action of renal buffers. Okay, so if this pH is not maintained by this buffer, this acidification will not take place. Acid-base balance by kidney is not taking place. So it is maintained by this pH. Minimum possible pH is maintained by the action of renal buffers. So what is the fate of secreted H plus? It is reabsorption of filtered bicarbonate. That is early part of PCT. That is sodium, potassium, sodium H plus counter transport in the early part of PCT. Then as phosphate, it is reabsorbed as phosphate because NaHPO4 minus combine with H plus to form NaH2PO4 minus. Dibasic phosphate is converted to monobasic phosphate and it is secreted as ammonium ion. So these are the fates of H plus which is secreted in the renal tubule and it maintained the pH of the urine. So there are three buffers in kidney which maintain the pH of urine. One is bicarbonate buffer, pK of bicarbonate buffer is 6.1. Other one is dibasic phosphate buffer, dibasic phosphate, monobasic phosphate. pK is 6.8 and ammonia buffer, pK is 9. These are the three buffers in our renal tubules to maintain pH of urine. Now bicarbonate. It is freely filtered by glomerular capillary in glomerular filtration. And the amount of bicarbonate filtered by kidney per day. How to calculate? That is concentration of bicarbonate in blood is 24 milliequivalent per liter. And amount of urine filtered per day is 180 liter per day. So what is the amount of bicarbonate filtered per day? That is 24 multiplied by 180. So we are getting 4320 milliequivalent per day. This much amount of bicarbonate is filtered per day. And reabsorption of filtered bicarbonate is necessary to prevent loss of base. 4320 milliequivalent is filtered per day, out of which 85% of filtered bicarbonate is reabsorbed in PCT and 10% is reabsorbed in thick ascending limb of loop of Henle. About 5% is reabsorbed in uh, collecting duct and DCT and collecting duct. Okay. So reabsorption of bicarbonate, 85% is PCT, 10% thick ascending limb of loop of Henle, and remaining 5% in DCT and collecting duct. Okay. So this is the mechanism. Sodium potassium antiport in early part of PCT is associated with bicarbonate reabsorption. Okay. So bicarbonate efflux across the basolateral membrane takes place by electrogenic sodium bicarbonate co-transport. In the basolateral membrane there is sodium bicarbonate co-transport, and an exchanger also takes place in some part of renal tubule, that is chloride bicarbonate exchanger. So there are two mechanisms of bicarbonate intake into interstitial fluid. One is sodium bicarbonate co-transport. Other one is sodium bicarbonate chloride exchanger. Okay. Now what happens in alkalosis? In excess alkali in our body, so serum bicarbonate increases. In case of alkalosis, serum bicarbonate will be more, and more bicarbonate will be filtered, and excess bicarbonate in tubular fluid than H plus. So amount of bicarbonate in the tubular fluid will be more than H plus concentration. Okay. So all the filtered bicarbonate cannot be reabsorbed. So some amount of bicarbonate is excreted in urine. Okay. And urine will be alkaline, and this help to correct metabolic alkalosis. Okay. If excess amount of bicarbonate in our body, it is excreted in urine. Now what happens in acidosis? There is excess H plus related to bicarbonate. Okay. So bicarbonate reabsorption is associated with H plus secretion. H plus is excess, so all bicarbonate is reabsorbed because bicarbonate reabsorption is associated with H plus secretion. So all bicarbonate is reabsorbed, and excess H plus is pass into the urine. Okay, so excess H plus pass into urine without bicarbonate reabsorption, and excess amount of H plus is excreted via urine. So acidosis is corrected. Now, phosphate buffer. We are discussing about phosphate buffer. In the distal tubule, this phosphate buffer acts in distal tubule and collecting duct, because the phosphate is concentrated in distal tubule and collecting duct because of water reabsorption. So main site of action of phosphate buffer is distal tubule. Okay, so this H plus combined with dibasic phosphate. H plus combined with dibasic phosphate, and monobasic phosphate is formed, and pK is 6.8. Okay. As a result of this action, a new bicarbonate is generated. Okay. So this is the mechanism of action. It mainly occurs in the interconnected cells, distal tubule and collecting duct, interconnected cells. Okay. So there will be H plus secreting ATPases, sodium H plus ATPases. Similarly, sodium, potassium H plus ATPases. So this H plus is secreted with the utilization of energy in the form of ATP, and this H plus combined with dibasic phosphate. H plus combined with the dibasic phosphate, as a result monobasic phosphate is formed. Okay, and this carbon dioxide inside the cell combined with water to form carbonic acid. Okay, it again split into HCO3 minus and H plus. Okay, so this H plus is secreted and bicarbonate is taken into interstitial fluid. Okay, So a new bicarbonate ion is reabsorbed as a result of H plus secretion by this mechanism. Now titratable acidity. So what is titratable acidity? It is the amount of alkali that must be added to urine to return its pH to 7.4. It indicates the pH H plus concentration of our urine. It is the amount of alkali that must be added to urine to return its pH to 7.4. Okay, it did not indicate the H plus concentration of our urine. Now, ammonia buffer system. So, ammonia buffer system, it occurs in proximal and distal tubules. So, what is the main source of ammonia? It is glutamine. Glutamine is converted into glutamate by the action of glutaminase, and one ammonia NH4 is formed. Again, glutamate is converted into alpha ketoglutarate by glutamine dehydrogenase. As a result, again one ammonium ion is formed. And by the metabolism of alpha ketoglutarate, two bicarbonate ions are also formed. Okay, so totally two ammonium ion and two bicarbonate ion is formed from a glutamine. So this is in proximal tubule. This ammonia exchange with sodium, ammonia sodium exchange in the PCT, proximal convoluted tubule. So ammonia is secreted and sodium is reabsorbed. As a result, two molecules of bicarbonate ion is reabsorbed into renal interstitium, is taken into renal interstitium. Okay, it decreases acidity of blood. So because two bicarbonate ion is reabsorbed. And in collecting duct and DCT, there is no mechanism to reabsorb ammonium ion, but all ammonia can be easily diffused across the tubular membrane. So there is no mechanism of reabsorption of ammonium ion, so it is converted to ammonia. NH4 plus gives to NH3 plus ammonia. This ammonia is freely diffused. Okay, and H plus is secreted by H plus ATPase. Okay, and inside the tubular lumen, this Na3 combined with H plus form to NH4 ion, right? And once it is converted to NH4 plus, it is not reabsorbed back, right? So it is trapped here in the tubular lumen. So Na3 plus H plus in the tubular lumen gives NH4 plus, and it is trapped in the tubular lumen. It is called non-ionic diffusion. Okay, it increases in extracellular H plus stimulate renal glutamate mechanism. Okay, if extracellular H plus concentration increases, it increases renal glutamate mechanism metabolism. Okay, now factors influencing acidification of urine. It include it depend upon bicarbonate load in our glomerular filtrate. So if bicarbonate load is more bicarbonate is reabsorbed. A kidney try to reabsorb more bicarbonate, and it is associated with acidification of urine. And increased PCO2, that is hypoventilation, partial pressure of carbon dioxide will be more. So more bicarbonate, as a result, more bicarbonate is reabsorbed in order to overcome respiratory acidosis. Decrease extracellular pH. If extracellular pH is decreased, so what happens if extracellular pH is decreased? That means acidosis. Okay, so more H plus is secreted. Now, role of angiotensin 2. This angiotensin 2 stimulates sodium H plus exchanger in PCT. So it increases it this role of angiotensin 2, it stimulates sodium H plus exchanger in PCT. Okay, so it increases acidification. Then aldosterone. It stimulates H plus secretion by intercalated cells in the collecting duct. Okay, so there will be alkalosis in Conn's disease. Conn's syndrome, there will be alkalosis because of this mechanism. Now, what is renal tubular acidosis? It is renal acidification is impaired, right? So, what is renal tubular acidosis? There will be acidosis in blood due to renal cause. That is renal tubular acidosis. So, renal acidification is impaired. Means urine will be less acidified. That means H plus concentration in blood will be more. That is renal tubular acidosis. Acidosis in blood due to renal cause. A type of metabolic acidosis. There are two types of renal tubular acidosis. That is proximal renal tubular acidosis and distal renal tubular acidosis. Proximal renal tubular acidosis. It is a defect in proximal tubule H plus secretion. So proximal tubular H plus secretion. So there will be decreased reabsorption of filtered bicarbonate. Okay. So the excess base is lost in urine. H plus secretion is affected. H plus will be less in urine. That is proximal renal tubular acidosis. As a result, less acidosis in renal tubule. Excess acid will be there in our blood. So this decreases bicarbonate, plasma bicarbonate, because excess bicarbonate is filtered bicarbonate is not reabsorbed. So it decreases plasma bicarbonate. Okay. So decreased plasma bicarbonate is metabolic acidosis. It leads to metabolic acidosis. So main causes are Fanconi syndrome and administration of carbonic anhydrase inhibitors drug. Now distal renal tubular acidosis. It is due to defect in distal tubular H plus secretion. It is due to defect in intercalated cells.